Control of gene expression in eukaryotes. In case of eukaryotes, gene expression can be controlled at different points. It can be controlled at the level of transcription. It can be controlled at the level of post-transcription, at the level of translation, and at the level of post-translation or post-translational level. Control of gene expression in eukaryotes. Here, the phenomena is that DNA is converted into RNA. In other words, it is messenger RNA. Then this messenger RNA moves from the nucleus into the cytoplasm or cytosol. Then this messenger RNA produces a protein. Now, where are the points where gene expression can be controlled in case of eukaryotes? Gene expression can be controlled at the level of transcription when DNA is going to convert into RNA. It can be controlled at the level of RNA processing when there is RNA editing or alternative splicing. It can be controlled at this point. When there is a movement of messenger RNA into the cytosol, RNA transport can also be controlled. So translational, when messenger RNA is going to translate into protein, the gene expression can be controlled at this point. And once the protein is produced, it can be inactivated. So gene expression in case of eukaryotes can also be controlled here. So these are the checkpoints where gene expression can be controlled in case of eukaryotes at the level of transcription, at the level of RNA processing, at the level of RNA transport, at the level of translation, or once the protein is produced, it can even be degraded by the microRNAs. Control of gene expression in eukaryotes can be by chromatin modification. It can be due to the chromatin modification. DNA is methylated, histone is acetylation. Control of transcription, alternative splicing, degradation of messenger RNA, and blockage of translation. So these are all checkpoints where the gene expression in case of prokaryotes can be controlled. A eukaryotic gene with control elements. Here we can see that this is a eukaryotic gene that consists of exon, exon, and intron. Here is the poly A tail. Here we can see that this is the promoter region of the gene, and here are the sequences which are called as proximal control elements. Then there are sequences which are called as distal control elements. Distal control elements can be in the form of enhancers or silencers, while in case of proximal control elements, they can be in the form of promoter, where there is attachment of the RNA polymerase or there is the attachment of the journal transcription factors. So journal transcription factors, they bind here and they control the expression, while the specific or special transcription factors, they bind in the region of enhancers or silencers. Control of transcription. Control elements, they can be enhancers and silencers. Transcription factors, that is DNA binding activators and repressors. Control of transcription, here we can see that here are the enhancers, that is distal control elements. We can see here that the promoter region is here and gene is here. So along with the gene, there is a promoter region where there is data box, and far away there are the sequences which are called
process. अब क्या होगा जो client
transcription of the genes. Another method by which eukaryotic genes can be controlled or their expression can be controlled is the alternative splicing. Alternative splicing is that one gene can produce
हम आते हैं तीसरे मेथड की तरफ
RNA, or it can produce more than one proteins. So gene expression in case of eukaryotes can also be controlled with the help of alternative splicing. Control of gene expression can also be due to the degradation of messenger RNA or the blockage of the translation. There are microRNAs that have been identified in the genomes. These microRNAs, they attach to the messenger RNA that has been translated by transcription and by transcription, this messenger RNA is actually either it is degraded or the blockage of the translation is blocked. With the help of microRNA can block the control of gene expression by two methods. First method can be that the microRNA, they degrade the messenger RNA. If the messenger RNA is degraded, it means there is no translation, there is no production of the protein from messenger RNA. And it can also block the translation when the microRNA, they will bind to the complementary messenger RNA, so they will block the translation. So by two different methods, gene expression can be controlled at the level that if microRNAs, either they degrade the messenger RNA or they block the translation. Control of gene expression can be in the form of post-translation. Post-translation can be the protein processing and the movement of the protein or the transport of the protein. Control of enzyme activity by effectors and inhibitors. Proteasomes degrade ubiquitin tagged proteins. So here is that protein to be degraded, that is the protein, ubiquitin, and there is a proteasome, and the protein entering into the proteasome, this protein will enter into the proteasome, and this proteasome will degrade the polypeptides. Proteasome and ubiquitin will be recycled, while the protein, this was the actual protein, this protein will be degraded into the polypeptides, or it will be no more functional. So this is another method that how genes can be controlled at the level of post-translation. So there are different control points in case of eukaryotic gene expressions. Those control points, they are chromosomal activation and deactivation, control of initiation of transcription, processing of the RNA, control of RNA transport, control of messenger RNA degradation, control of initiation of translation, that is usually in case of eukaryotes, and the gene expression can also be controlled by post-translational modifications.